Well, welcome everyone, and good night and evening, and all of the wherever morning, wherever you're watching this from. So, tonight we're going to talk about precision medicine, and we'll talk about it in the sort of clinician and layman scientist friendly way that we've been attempting to speak about these things. Um, so we'll talk a little bit about what it is, what we're already doing in practice, and why it's still hard to implement a lot of these things. Because a lot of times we've spoken about pharmacogenomics before, and a lot of times the big issue seems to be actually the implementation of it. So there are two ground rules, I would say, for these webinars. First of all, this is purely educational. Um, we'll talk about principles and workflows, but this is definitely not individual medical advice. And I am very much still human and able to make lots of errors. So, and when I do, by all means, please correct me. And please, I see some of the panelists and list attendees joining. So, welcome to correct me live, interrupt me, and you're also welcome to send me emails and comments. These do tend to be distributed on a few platforms. So, you're all more than welcome to correct me and send me an email. That's the one thing. So the second thing is, of course, that I'll define terminology as we go. So you don't necessarily need to sit with your appendix. So you don't need a genomics background. You don't need a precision medicine background. A little bit of a biology or clinician background goes a long way. Most of our viewers do tend to be clinicians, but I do think it should be friendly to everyone. So why this topic, precision medicine? Um, why now? So over the last decade, we've made a lot of progress in terms of precision medicine and personalized medicine. So in 2015, there were large national and international initiatives, sorry, announced to accelerate molecular tools and infrastructures needed for routine clinical use regarding or revolving around precision medicine. And the reason you feel it clinically is quite simple, actually. So we now have the ability to generate patient molecular data quickly. And the pressure is moving from can we do, say, genome sequencing to can we interpret it and use it safely. So I think an important distinction to make as we go through tonight is the distinction between precision medicine and personalized medicine. So let's define those clearly. So precision medicine is not necessarily an individualized molecular approach. So you examine a patient's profile, whether that's genomic or proteomic or the gene expression profile, so that would be the exome, and many other molecular biology markers, and you use them to select or develop different treatments. Now, the key idea is that precision medicine often groups patients or individuals into populations and subpopulations based on molecular profiles, and then based on these profiles, you assign different regimens uh, that would lead to better drug use or better drug implementation. So I'm talking about pharmaceutical drugs, of course. So no adverse drug reactions or no non-responders or that type of thing. Personalized medicine, on the other hand, is sometimes used interchangeably, colloquially, but the definition is actually a little bit more specific. So, personalized medicine is actually designing a treatment that could be unique to each individual um, based on their molecular profile. So, if you think about personalized medicine, it is precision medicine, but it's a subset under the precision medicine umbrella term. So I used a couple of words. They use genomics and transcriptomics and exome and all of those things. So let's just define those before we move on. So genomics is quite simply it's the DNA sequence and variants across the entire genome of an individual. Transcriptomics refers to which RNA messages are being made into proteins or and how much of it. This is all referring to gene expression in action essentially. Proteomics is what types of proteins are present and also how much. Metabolomics It's actually quite an important one when it comes to this sort of pharmacodynamic, pharmacokinetic settings. Metabolomics is a small, or what these molecules do and then how they interact um, in this case of medical intervention with the drugs to make the metabolic products. And metabolomics is actually also often closer to the phenotype that we see eventually than the DNA is alone. So yeah, so it's all about the small molecules and metabolic products that are present in the individual or population. And then epigenetics, we spoke quite a lot about epigenetics last week, and we'll actually dedicate a whole session to it in one of these coming weeks as well. So, but just the recap of last week. So, epigenetics are changes that affect gene expression without changing the underlying DNA letters themselves. So, this includes methylation, this includes histone acetylation, or also methylation, or any histone modification. Histone is of course a structure in the DNA. So methylation would, for instance, tighten that histone, and that would affect how genes are expressed as well. But the key theme tonight is that although the DNA sequence is foundational, so which is the key to genomics, it's not actually the full picture. It's not enough to explain what's happening clinically, although it is extremely useful to start off with. So I know we've done a whole episode on this as well, but let's also just talk a little bit about pharmacogenomics again. So, pharmacogenomics is probably the most developed part of precision medicine. Pharmacogenomics asks how does a person or how does a person's genetic makeup shape their response to a drug? It also includes using that knowledge to develop and supply completely new drugs with new targets, and specifically in the case of genetics, with different genetic profiles. So, pharmacogenetics is often used interchangeably with precision medicine, but it's actually also more focused and narrowed down onto how does a sequence variation in a specific gene affect a drug response, right? So, so yeah, let's look at that. So the first example there is optimizing drug response. So that could be very blatantly reducing adverse side effects or adverse effects, and then also trying to identify which populations are non-responders to drugs. So a lot of times when you do give these prescriptions without the pharmacogenomic testing beforehand, you find there is the potential to have non-responders. So that's the optimizing drug response pathway. And then the other pathway is, of course, developing drugs targeted to the specific molecular subpopulations. And I see we're joined by Professor Jeffrey. Stephen, how are you doing? Stephen's just connecting his audio. He'll join us audiographically in a second as well. In
Hi,
the
Chris.
meantime, hi, Stephen. How are you doing?
Yeah, I'm fine, thanks. Yeah. And you?
Yeah, yeah, fantastic, fantastic. So we were just talking about precision medicine and how it's distinct from personalized medicine, in that precision medicine is an individual's molecular approach or molecular makeup that defines how they react to different drugs, and this is often done by putting them into different subpopulations. Whereas personalized medicine is sometimes used interchangeably, but it's actually little bit more individual to that specific patient. So it's a subset of precision medicine. And we could define a couple of genetic terms like um, genomics and transcriptomics and proteomics, and epigenetics and so forth, which we'll talk about again as it comes up. And then we started speaking about a topic that we've spoken about before, which is pharmacogenomics and how it's, I would say, probably the most developed part of precision medicine, and how this is essentially divided into two groups. So it's either optimizing drug responses, so trying to alleviate adverse drug effects or select drugs appropriately so that you don't see these adverse drug effects, or sometimes non-response. Um, and then the other avenue is, of course, identifying new target populations or subpopulations, find drug alternatives that work with different gene targets so that that you can actually ascribe better medication to patients. And so why clinicians should care about variability and how this influences adverse side effects. Um, I'll actually give you a real world motivation. So there are serious adverse drug effects that can happen at scale, and many patients that don't respond as expected, which is not completely, but very, very largely influenced by genetic variation. Just to give some examples of mechanistically how gene variants can affect response. So you can, if you have a gene variant, this can affect things like carriers or transporters. So this is how the drugs get in or out of cells. It can affect receptors, so what the drug actually binds to. It can
Hmm.
affect the enzymes that have to act on these drugs, either by degrading them or modifying them. So modification would be, for instance, how codeine gets modified into morphine by the body. And of course, it also affects how proteins store or excrete these drugs. So, in other words, a drug response is not just the drug, but it's the drug compared to an individual's genetics is going to determine how the drug actually gets metabolized. And so I said I would give a real world example. So a very probably the most famous example is the cytochrome P450 enzymes. Um, Now, yeah, so this is a flagship example. So cytochrome P450 is a gene family, and these enzymes are all ones that metabolize quite a long list of different medications. So, and in a nutshell, if someone has the variants for a slow metabolism, the drug active product can accumulate, and that usually raises toxicity in the case of something like codeine or warfarin or any of those drugs. But the more dangerous case scenario is actually if someone metabolizes a drug too quickly, because then the drug can either be cleared before it works, or if it's a case where it's getting altered into a different active form, then you have too much of this drug. So, uh, CYP2D6 is an example of this cytochrome P450 gene family, and it's involved in metabolizing, I think, about a quarter of all commonly used pharmaceutical drugs. Let that sink in. A quarter of all is interacts with this one gene. So, and that includes examples like acetaminophen, clozapine, beta blockers, taxaminofen, codeine. So there's a lot of very common drugs that are affected by
So
CYP2D6.
that so sorry, so that's quite a huge proportion, and the fact that we don't even bring that into our equation or into our prescribing concepts at all is quite remarkable. And
It is, yeah.
yeah, so I mean, it's and do we do you know the extent to which this is, or is it that it impacts on the outcomes of the drugs in terms of adverse events and efficacy?
So I know not for all drugs, but I know for some. So for instance, for CYP2D6, there are different metabolizer categories, and most people, around 80%, are the term for it is extensive metabolizers. So that means they either have one or both of the normal wild type genes, so they kind of react to medication as we expect them to. But about between 10 to 15% of people are poor metabolizers. So that usually means they have two copies of a gene from each parent, or one copy from each parent, that has decreased in activity of what that gene needs to do. And the remainder of patients usually have something called duplications. So they have duplicated genes, and they are then described as these ultra rapid metabolizers that break it down extra fast, or the so essentially, with that in mind, so about 80% of people will not be affected necessarily by the genetic profile in the drug, but the other 20% will be affected in some way, either by breaking it down too much or not breaking it down, metabolizing it properly. And clinically, this explains a big pattern as well, because one patient gets a side effect at the normal dose and another gets no effect at the same dose.
Yeah,
Yeah.
yeah. So it's quite profound. How easy is it to test for that the effect of that sort of, I don't think you can call it a mutation, but that gene on the,
Yeah.
can, is it something that we can test and apply clinically quite easily, or will that take some time for us to do?
So it can be done, and it is actually done in a couple of places. Last week we listened to a talk, or you hosted a talk with Professor Ishmael Borad, and he was also talking about the incorporation of these gene tests. So what we're seeing a lot of these days is there's an operational shift moving from reactive testing, so that's ordering a single gene test when you need a specific drug, to preemptive pharmacogenomic screening. So in the sort of pediatric program where broad testing is done upfront and only clinically validated results are surfaced to the clinician. This is an example of this. So as the evidence grows, we get these additional sort of gene drug alerts, and we add them to electronic records. But as you know, it is something that we can do, but it isn't something that we often do. And I
Mm.
think a lot of times when you look at single genes, and in terms of how easy it is to do this, the people have in the past always focused on what's called the high yield gene drug examples. I'll give you five of them. So the one big one is the TPMT variants. So just medically, TPMT enzyme levels can lead to toxic side effects with drugs like thiopurine, acatapurine, that type of thing. So the TPMT variants help or affect how the enzymes break down these drugs. Another very famous one is the HLA-B alleles or genes, and these are associated with all sorts of allergic reactions to certain drugs, um,
Mm.
including allopurinol, abcavir, carbamazepine, mezapine. So this is another very relevant high yield drug example. That's a fairly easy test because you know the drugs and you know the gene. Another drug gene interaction is warfarin. So warfarin actually has two genes that are highly associated with it. So the one is called Vickers or VKORC1, which is
Mm.
the drug target, and the other one is called CYP2C9. So it's one of those cytochrome P450 ones that I spoke about a little bit earlier. And these variants can usually lead to overdose effects at the typical dosing. So people know how to scan for TPNT, HLA-B, Vicker or VKORC, and CYP2C9. And then the cytochrome actually have a few. So there's also the one called CYP2C19, which affects the metabolism of a significant subset of drugs and spores, and spans from everything to poor to ultra rapid metabolizers. So, and there's actually another important one called SLCO1B1, which is it has to do with how the liver transporter affects statins. So variants can reduce the removal or increase the exposure to removing statins. So yeah,
Sure.
and these are fairly easy to test for. You can test for them with a regular PCR. The problem is with the ease of testing is not necessarily the literal complication of it, but it's the fact that if I, if I if I go to a clinic and I want to have a PCR machine and I want to do a single gene test, the only way I can do that in my own clinic is if that clinic also has an ISO registered laboratory.
Mm.
Otherwise, what I have to do is I have to send it to a service provider that happens to do this single gene variant, where some places are fortunate enough to have those. In Cape Town, there are some people who do things like that. In Durban, we have a lab that also does a couple of sort of genetic based screening tests, but they're not exceptionally abundant, especially in developing countries. You don't always find these gene tests. Um, and so with that, the answer to how easy is it to implement, how easy is it to do, is all a completely relative one in terms of where are you located and what are you lucky enough to be in some place like Cape Town, which has a lot of resources in the surrounding area, where you can actually do this type of thing.
So, but I mean, I must say, for some drugs, I mean, they are chronic drugs, and a once-off drug like codeine or whatever, patient is got pain, it might not be so relevant. But you think about a patient who gets diagnosed with a pulmonary embolus, or maybe one of the thrombophilias, and would need to go into something long term. And
Absolutely.
it might ultimately make the biggest difference to the efficacy of that drug and ultimately, increase their survival by being
Yeah,
able to test it, and also
100%.
helping dosing, etc. So, that's why we do sometimes struggle with patients who are on warfarin and their INRs all over the place,
Yeah,
and
yeah.
you struggle. And so, I think warfarin is jumps to mind immediately as the one that I think would be very useful. Are there any other drugs that you immediately think could be where this would roll out very quickly?
Yeah, well,
Uh,
warfarin actually is also one of the first ones that I think about because it causes central nervous system bleeding in an overdose.
yeah,
But
yeah.
codeine, there have been examples of breastfeeding mothers that are given the standard dose of codeine, and then because their bodies break it down fast, this actually affects the infant. Some
Yeah.
infants have
Look,
died.
I mean, I think it would be great for everybody to know their codeine and all sort of the um, morphine type
Yeah,
um, metabolites. Do
yeah.
you think does that all work down the same pathway? Morphine derivatives. I mean, there's so many these days. You've got Targinact, you've got Oxynorm. You've got these derivatives and your fancy ones. Got different absorption methods. Do you think we can somehow get to the bottom of that and or do you have to test for morphine and all different variants of that metabolizers?
A lot of these drugs have a similar biological pathway, or at some point link up to a common biological pathway. So if you, for instance, if you look at psychiatric drugs, anything that's used to prescribe to ADHD, for example, will at some point probably link into the amphetamine breakdown or metabolism cycle. So there are definitely underlying biological pathways, and these are ultimately going to be affected by gene transcription factors and different genes that affect how different enzymes in these pathways interact and act on drugs. So there's definitely, and I think that was the thinking between with the high yield genes in the past, because people said all of morphine drugs do go through this pathway. This pathway is most highly affected by this gene. Let's only look at this one and variants in this one. But that's where electronic records and more importantly whole genome sequencing sort of changes the game, because the clinical promise there is efficacy and efficiency. So instead of ordering these separate tests for these separate genes every time um, that you're about to prescribe a high risk drug, or often just want to avoid adverse side effects, you already have a pharmacogenomic baseline on file, right? So you do a screening once in a person's life, and you already know, yeah, you know the high yield ones as well, but you also have all of the low, or the ones that tell you something but less. So uh, yeah, and we are definitely heading in that direction of actually having all of that stuff um, on file.
Would so and then just to come then down to the role of the whole genome, would that pick would if you had that on some sort of database, would you then be able to cherry pick from your genome some of the genes that you would want, or would that pick it up automatically? How does it work from the whole genome testing?
Yeah, so the you if you have, let's say you've done a genome sequencing and you have your genome on file there, then there's multiple different approaches. You can take either a top down or a bottom up so approach. And so let's say a patient comes to you with a certain disease phenotype, you can work that way around and say, okay, let's work back from this, which pathways are involved, which genes are involved, are these also connected to a drug? Or you can do it from the side of, I want to prescribe this drug type to a person. Let's see which genes are involved, which pathways are involved, etc. So you can do it either or, and you asked if it would pop up. So uh, with these days with machine learning type things, you get the term that they use for it, it gets flagged. So yeah, you do actually see these flaggings in the
Hmm.
in silico analysis, and it says this pathway is likely or probable to be involved. And yeah, so you do actually see that on the computer. So, but then from there, it's all statistics, right? So stats is never a defined definite. So it could be linked, but you don't know. All right. There's a lot of these, especially when you're talking about genome sequencing, there's a lot of genes that seem to be identified, that seem to be connected, but no one has ever done actual causatory or causation analysis. So no one's physically gone and seen if I mutate this gene out and give this drug, does it affect it? So we can't claim causation. We can only claim probability, but it does pop up, and even more so these days with our fancy machine learning tools.
But ultimately, it might actually just be good to know where the flags are. So if you had a choice of drugs and you know that one is flagged, then you would immediately default to the other one. So that's,
Exactly.
you know, a place where it would be useful from the beginning. Um, so we've spoken about the morphines. We've spoken about the warfarins. Is there any other class of drug we felt that this might actually be really useful? What about the
Ye-
antiretrovirals or antihypertensives, diabetic drugs? Those are the things that really are very commonly prescribed. Diuretics, cholesterol
Yeah,
drugs. Are we heavy
absolutely.
on that? Because I mean, half the population's on that.
Yeah, all of the above have some level or another of genetic regulation, or genetic influence. Some, so a lot of times when they do these sort of studies, and they tend to first focus on the diseases or disorders that have a high genetic background, so cancer, for instance. But all of these diabetic diseases, metabolic syndrome, the antibacterial, and is actually another. So your anti precision medicine based antibacterial resistance, um, and which antibiotic is going to induce a certain something. Are you going to be penicillin uh, allergic, for instance, is another
Yeah.
big thing. So yeah, all of the above have gene variants that we know of that do influence the drugs um, capabilities of being metabolized by a specific individual's body, but not always implemented in terms of screening. Because if I know that, say, malaria drug X is affected by this gene, but it only affects about 5% of people, and in those people that are affected, it's only about 10% of the story that's actually dictated by genetics, and the rest is lifestyle and diet and nutrition and so forth. Then it becomes less and less sensible to do a test for that one specifically. But that's why a lot of clinicians and scientists are pushing this idea of, but let's do a whole genome or exome sequence, because then we have this information. Cost wise, it makes sense. And then if we have the information, then let's look at it, because it does inform us on better decisions for which drugs to do it for. Quite literally, all of the examples that you listed there. So, and I'm actually going to give a perhaps a too extended example of targeted drugs um, on precision oncology, so cancer. Um, and I've always found it interesting how, if you look at this, there's a massive disproportion of studies that are focused on cancer, as opposed to other drug theory. But it's a lot of times what happens in the field of oncology and cancer is actually a little bit of a technological pioneer for what we eventually do use in other fields. So I'll give this example. So precision medicine direction was developing drug targets to molecular subpopulations in cancer studies. And what they did essentially is they looked at something called HER2, HER2, right? So HER2 is a gene that codes for transmembrane receptors, and it involves signaling that promotes growth and divisions of cells. So in a certain subset of a population um, of invasive breast cancers, about 25%, so another quarter, uh, have this HER gene variant, and which makes it amplified, and the protein is completely overexpressed. And so this status, this overexpression, is associated with more invasive more invasive breast cancer and poorer prognosis. Now, on the other side of that, you have Herceptin, which is a monoclonal antibody that binds to the extracellular region of this Her receptor. And what it does is it inhibits cell signaling, it triggers cell cycle arrest, and it leads towards this cell destruction and of the cancer cells, so apoptosis of cancer cells. So um, what they do is they find these people with this specific drug, um, and they do target testing. So there are two ways to do target testing. So you get one is called IHC, immunohistochemistry, which is an antibody that binds to this HER2 protein in our specific case, um, in fixed tissue, and then the staining shows the overexpression visually. And then the other thing is called FISH, FISH, and it's fluorescent in situ hybridization. And so what it does, it compares fluorescence from a HER2 DNA probe with a control probe to assess for amplification. So this is how you identify the people who are part of that 25% who have the variant. Um, and then once you know who has these, you can actually prescribe a specific treatment, that aims to this target population, that detects what is the tumor expression of this population, and you create a drug that is actually catered towards this population and is actually much better at treating them. So it's a classic example of being able to identify a variant with a known much better drug alternative, and to employ precision oncology to then treat this, which would have otherwise been a much more rapidly developing type of cancer. And there's actually another example of that where they use what's called EGFR antibodies plus the KRAS system, KRAS, which is part of the P53 cycle. So, and this is in colon cancers. So EFGR targeting, monoclonal antibodies help a certain subset of patients, um, but where the cancer cells must be expressed by the EFGR, and then the wild type, they call it the KRAS or the KRAS gene. Um, and so sometimes the biomarker isn't enough it can't just be present. It also needs to affect the downstream pathway. But when they do have the two required things, it can be detected by a simple PSCR based method. And then we can once again employ this sort of precision oncology immunotherapy to try and target this specific population where we know the combination of this drug and this phenotype leads to a much better treatment of it. So yeah, it's quite interesting. So how precision oncology actually aims to target these mutations and expression defects, in tumors, and then to create these much more effective remissions, and potentially even cures for some cancers. But of course, and nothing can be, when it sounds too good to be true, it is too good to be true. So there are difficulties with it, which it includes each tumor is genetically unique. So this is the difference again between personalized and precision medicine. So each individual is going to have an individual genetic composition in their tumors, and there are large research efforts that are trying to map out sort of whole tumor genomes and trying to identify how these individual profiles can also be targeted. Um, and the other big problem is, of course, that only about 6% of patients can be matched to a targeted drug based, um, on a tumor genetic profile. So it's not a very large amount of people that will actually benefit from this type of treatment. The other big thing is, of course, tumor resistance, is a major issue. So because as the layers of tumors develop, this inherently how cancer works is with each layer you get more and more mutations. So you can't actually just use immunotherapy. You need a combination strategy, with targeted and generalized approaches to circumvent especially cancer resistances. Um, sorry, Steve, I can see you're speaking, but you're muted.
Sorry about that. Absolutely. So it's amazing that this is now really transforming the whole world of oncology and oncology treatment. How many of these treatments are actually rolled out into standard practice? Because we've been talking about these things for quite a long time now about the use of the gene therapies. But
Yeah,
what is available? I mean, obviously the most common cancers, breast, colon. Um, where are we in terms of having these really available in a place that's relatively well resourced, or is it
yeah.
still
So
expensive in most places?
no, there are so some of the most promising ones in cancer immunotherapy specifically, The one is called ACT, adaptive cell transfer, and the other one is engineered T cell methods. Specifically, they look at CAR T cell therapies. So, like, to make sense of that, we kind of need to just take a step back and remember something from the immune system. So, the innate immune system acts rapidly and non-specifically, and the adaptive immune system is specific and has, they call it a memory, right?
Mm.
So adaptive immunity has two branches. So the B cells produce antibodies, that's humoral immunity, and the T cells mediate cell-based immunity. So especially important for recognizing and destroying infected or cancerous cells. So if you think about sort of the useful terms in terms of that, so antigen is a non-self marker that the adaptive immune system can recognize, and the something called the antigen presenting cells, so the APCs, um, process these sort of protein fragments that present them. So if you have T cell receptors, um, or TCRs, you are able to have the T cells recognize specific antigens presented in what's called an MHC molecule. So these are just cell surfaces that 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 where the T cells bind to the fragments of the antigen. Um, and then this what in short is what happens then the cytotoxic T lymphocytes or CTLs come and they recognize these cells and they kill them via the release of all sorts of molecules like perforin and granzymes. And they essentially kill the cells through apoptosis. So yeah, so that's uh, how the adoptive cell transfer, the ACT method works. Um, but there's something called neoantigens and TILs. So this is why or how they're able to actually make that tumors are recognizable. So uh, cancer cells are often express proteins that are recognized as non-self because of mutations and abnormal expression. So the body recognizes this is something different, something that's being expressed now, it shouldn't be. And a lot of times these novel targets are called neoantigens. So T cells often infiltrate tumors. So that's a TIL, so tumor infiltrating lymphocyte. Um, but tumors also have the ability to suppress T cell functions using all sorts of strategies like inhibitory molecules, that repress the T cells or altered MHC expression, regulatory T cells or Tregs that dampen the activity. So this all sets up why the methods like the ACT and the CAR-T are actually needed so their attempts to sort of overwhelm or bypass this tumor suppression tract. So yeah, those are two examples in cancer immunotherapy specifically that are being used. So I can elaborate on ACT as well. So that's adoptive cell transfer. So it's more or less the same thing that it does, but what it does is it it um, also recognizes these tumor antigens. But the difference is you actually remove it from the patient beforehand and you amplify it in cultures and then you reinfuse it back into the patient. So it includes culturing a small tumor sample um, and selecting which effective T cells or T cell clones are the best and expanding them in high numbers and then reinfusing them into patients. So to reduce T cells or to reduce immune cells that suppress the activated T cells. So all of this is essentially an engineering approach, or genetic engineering approach to use a person's own immune system to try and target cancer treatments. The CAR T cell therapy is another one where they also do the same thing of yeah it's all more or less the same procedure. So they take in that case they take an extracellular antigen um, built from immunoglobulins or globulins with variable regions like antibody specificity, and then they use a transmembrane region to anchor it, and they once again do do an activation of T cells, which triggers proliferation and cytokine production and apoptosis of cancerous cells. So all of these are engineered genetic engineering ways of getting a person's immune system to target their own tumor cells, either by injecting something into the person or taking it out and reinfusing it, and ultimately activating or upregulating different uh, different immune related responses.
Oh. So it's quite profound, because if you think about medicine over the last 100 years, we've made progress in infectious diseases, and that is arguably been the reason for the reduction in early death. So that is maybe why we've had longevity's been so extended. But in some of the other much bigger ones, cancer, cardiac disease, Alzheimer's, diabetes, we've not really seen much in terms of a rapid development and progress.
Mm.
People still die from cancer all the time. They die from cardiovascular disease. And it's great now that we kind of have a little bit more of a little bit of a pipeline
Yeah,
for, for cure, even.
yeah,
Um,
yeah. Some
now that we
cancers.
understand a lot more about the genetics and we have these genetic therapies. So, do you think ultimately, if we did get to that holy grail of a cure for the various and some of the most common cancers, it would be along these pathways that you're talking about?
Yeah, I think so. You know, so genomic expression matters a lot. It's going to inform us a lot. And just to put into perspective, just how much we've actually developed in terms of screening and genetic tests. I think there are more than 45,000 genetic tests available, right, for different clinical applications. So
Oh.
there are a lot of different tests being used, but it can never be, or not never, but it is unlikely to ever be the full story. So the genome sequence alone won't tell you the whole story, and it's because phenotypes, so how things get expressed, depends on what genes are being expressed, how RNAs are processed, what proteins are present, metabolic states, and often dynamically and over time. So precision medicine's future, to answer your question, is not exclusively going to be sequencing to lead us to a cure, but it is integrating sequencing with a systemic sort of biological readout. Um, and I can actually, so if I think of a proof of concept, right? So repeated blood sampling um, over time, so over a patient's life, paired with a genome sequence, or transcriptomics, or proteomics, or metabolomics, um, can essentially give you so much information on terms of how to treat a patient throughout their entire lifetime, that the point doesn't become everyone should get this done now because it's going to inform any possible future medical intervention. But the point is more all of these modern tools, whether it's genetic, which is my field, or all of these other chemical and molecular tests that are being developed, um, may either now or in the future support um, earlier predictions or tailored treatments, or eventually perhaps even cures, but not in isolation. So it's all. Yeah,
And you touched on it a little bit now, but I suppose one of the other big discussions in the whole world of oncology is screening. And do you think
yeah,
we would be able to take things upstream a bit and screen better for some of the bigger cancers like colon, um, again, breast,
yeah,
lung, bone?
absolutely.
What do you think?
Absolutely. Yeah. So, yeah, because primarily the screening tools that they use for those now are single gene. So everyone has heard of the BRCA gene, the BRCA, and that's a high yield gene, and it does give a pretty clear indication of a risk for breast cancer being shifted upwards or downwards. Um, if you have the wild type homozygote, and you also see different populations. So, for instance, the Afrikaner population have a higher incidence of the BRCA gene. And so you know that that population tends to have a higher risk. And although these screenings are extremely useful and it help us make informative treatment options and treatment decisions, even just those single gene variants, so the ones that we already do, have all sorts of technical and social and ethical challenges that come with them. So generating data is getting easier and easier, but storing it, interpreting it, assigning meaning to it, it's still
Yeah,
hard, even with machine learning.
very
Um,
difficult.
yeah,
Yeah, I mean, a simple thing of doing a BRCA and then finding a positive. It's nice to know that it's negative, but the minute it's positive, what do you do with the
what
information?
do you do? Yeah, exactly,
There's been
exactly.
a lot
Because
of media attention around this as well.
yeah, yeah, I've heard, I know of some fairly famous people that got mastectomies
Yeah,
simply because they got a risk for BRCA or, or,
yeah.
or
I
breast
mean,
cancer.
I've got patients, I've got friends of family members. They um, did that you decide what to do, and it's very
Yeah,
difficult. But ultimately, we mustn't lose sight of the fact that these are life-saving tests, and
yeah, of course.
that's help people to know these things. But that's where we're going to have to bring in a whole army of support to help us interpret these, manage them, and I think obviously these conversations do need to be had more widely um, in medicine and in the way in the
Absolutely.
clinicians.
Yeah,
Uh,
medical education changes everything. Clinicians will increasingly be expected to interpret and explain genetic data, meaning we need more genomics literature for clinicians and more genetic counselors and specialists involved in this whole pipeline.
yeah, and it's something that we have spoken about before on a previous webinar. But the societies, the guidelines, the protocols will need to feed this genetic data in at some stage as well,
Yeah,
and
well,
it would need to be embraced. That I don't know. Are you aware of any societies that are bringing these into their protocols
there's
or
such a difficult social layer to this. So you always have to ask the question, okay, who pays for genetic tests and counseling, especially before symptoms? So if it's a preemptive test as opposed to a reactive test. That's hard enough with some of the screenings that we have. A lot of a lot of aids actually only cover something after diagnosis, but not necessarily simply having a risk. And so, how do we ensure that these tests actually are accurate and that the privacy of the genome data is stored in databases and healthcare records? Uh, and of course, the other big thing is how do we avoid widening inequality? So, if some patients can have access to expensive specialized care, who's to say that some patients won't have access to better genetic screening options or once they have these informative markers to that they are able to then so I'll give an example. So let's say you do have a high risk for a certain cancer, but the treatment of that is exceptionally expensive. Is it then ethical knowing that financially this person will not be able to afford the treatment for it, and there is no company or third social party that is going to pay for it. Um, so yeah, genomic or genetic discrimination, and
yeah,
you'll see this in employment and especially in insurance. Absolutely, this will come out in
yeah,
insurance.
yeah.
Um,
And that's
yeah,
most likely why we have tread very lightly over the last few years on some of this stuff. Because, and I kind of think it's a tip of an iceberg that a lot of people just don't want to go down because they just know that from there it's the expectation and the responsibility is going to broaden a lot because you're going to have to put in place all sorts of infrastructure to make sure that it's done properly.
yeah,
Yeah,
exactly.
but
And so it is a case of like, so who are the people that are going to take that responsibility? Because it's a major responsibility, and a lot of
mm,
things can go wrong. And so there is a lot of societal
mm.
reasons uh, for societies to not want to necessarily start with it, you know?
That's why I like the whole idea of the pharmacogenomics because I think it's more readily implementable. It doesn't have as many implications from a social point of view,
Yeah,
and it's very useful clinically. So, um,
absolutely.
yeah, no, it's Yeah, again, amazing thoughts, and we really continue to look forward to what next our colleagues will bring up. But it's very exciting to know that there is potentially some real hope for patients on the horizon for things like cancer.
Yeah,
Because to cure cancer in the past, you had to use a knife, but now you can use some genes possibly, and I think that's really nice. It's
yeah,
and
absolutely. We've
that
come a long way because yeah, we started with radical mastectomies. For instance, in the case of breast cancer, where you not only do you surgically cut out the small piece of cancer, but you cut out this entire surrounding area. That
yeah
eventually we developed radiotherapy. We eventually later we got chemo. Well, actually we got chemotherapy first, and then radiotherapy.
yeah
And now these days we're bioengineering the immune system to take care of the cancer itself. So it's
yeah.
quite remarkable actually how
Amazing.
far.
Yeah.
Yeah, yeah,
No,
yeah.
it's incredible. Yeah. No, thanks. Um, yeah, was there anything else you wanted to add tonight, or should we?
No, that's the end. So if you take nothing else away from tonight, you can take away that precision medicine is fundamentally not necessarily just about using molecular profiles, but it's what we use to make decisions in terms of prescribing different drugs, by stratifying people into different patient groups. Um, but ultimately, it's all about specific people benefiting from specific therapies by tailoring those therapies more individually. And this can be used in cancers. This can be used in psychiatry, and the future is not just DNA sequencing, but it's an integrated approach between integrating all of this data in the healthcare infrastructure with all sorts of ethical safeguards and societies that actually are willing to speak about, educate, and fund these things.
Yeah, it's
Yeah,
great. I mean, that's a great summary. Thanks so much. And
yeah,
Christian, great, another great session. Thanks so much for all your inputs here. And yeah, we look forward to the next one. Thanks so much.
absolutely. Thank you so much for joining. See you at the
Cheers.
next one.
Cheers. Bye. Thank you. Bye.
Cheerio.